Okay, thank you, Chair. Good morning, everybody. I'm going to talk about the potential of molecular biotechnology to improve disease control. I want to try and give you a feel for what we do in the lab to support industry and government. So maybe stating the obvious, why is animal health important? The Food and Agriculture Office of the UN estimates globally that by 2030 we're going to need to produce 50% more food to feed an increasing human population. Industry is driving to improve efficiency and productivity, and the FAO and the Animal WHO estimate that livestock diseases reduce the efficiency of animal production by about 20%, with significant economic and animal welfare impact. So an animal health problem is a production problem. Now, the challenges in disease control and the area where AFBI has a key role locally. Firstly, in epizootic risk, these are diseases, damaging and devastating diseases that we don't have and we don't want. Guarding against their introduction, and for AFBI providing an emergency response capability, examples being highly pathogenic avian influenza, blue tongue virus, and devastating epidemics of foot and mouth disease in 2001 are in our recent memory, and AFBI responded to those emergencies. We also are vigilant about new and emerging risks. Nature has a habit of throwing up new variants and new pathogens all the time, and the same is true in human medicine. There are new pathogens in veterinary medicine, especially viruses, things we'd never heard of, Schmallenberg, post-weaning multisystemic wasting syndrome associated with porcine circovirus. And in the human field, MERS and SARS are things that were unheard of just a few years ago. So 60% of all known pathogens and 75% of emerging pathogens are zoonotic in nature. They have that animal-human interaction. We're concerned about pathogens with increased virulence, which can come about through recombination, for example, between swine and poultry flu viruses, or as viruses tend to do, when they jump species, they can increase their virulence and become more pathogenic. We also have a role in supporting public health and food safety, and a big One Health issue of the moment is the predictable emergence of antimicrobial resistance in bacteria. And citing the Chief Medical Officer in 2011, global action is needed to tackle the catastrophic effect of antimicrobial resistance, which in 20 years' time could see any of us dying following minor surgery. So really troubling issue there. Climate change and the changing vector pattern, particularly of insect-borne viruses, allowing disease introduction and spread is a concern. And last but certainly not least are the endemic diseases, the big diseases that we do have and we don't want. And a classic example is bovine TB, costing Northern Ireland 317 million in a 15-year period to control, with major impacts on farming family welfare, and that's a major concern. So I said I was going to talk about molecular biotechnology, and I'm going to talk a little bit about DNA and the enabling technologies around this area and the tests and that have flowed from developments in this area. I'm going to talk about DNA, which is in the news all the time, and any of you that watch crime scene dramas are very familiar with it. It's the heritable genetic code of life, and it's written in chemical letters. This iconic and beautiful double helix of DNA, the structure was solved in the 1950s, and it led to the emergence of a new discipline called molecular biology, where scientists are able to go in and read some of that sequence and work out what it does, and develop tests and tools around the genetics of animals and plants and pathogens. And I want to talk about how AFBI deploys the techniques that have flowed from this. And sort of more recently, there's been further development and significant future potential in a discipline called genomics and bioinformatics, which is really genome data. And the genome is the genetic code of an animal or a microbe. And just to give you some scale, a virus such as the foot and mouth disease virus, its genome has 8,500 letters. The TB bacterium is a bit more complex, is 4.4 million letters. And the bovine genome is about 3 billion letters. It's similar in size to the human genome. And in terms of drivers and enabling technologies, the big developments in the last few years have been around DNA sequencing. We've been able to sequence and read that code or bits of that code since the late 90s, and what it has involved is reading the genetic code of animals and pathogens, working out how similar and how different they are, their gene content, information content. And to do that, chemistry was very labor-intensive and expensive until quite recently, and there's a game-changing technology called next-generation DNA sequencing, which, through staggering developments in chemistry and reductions in cost, facilitates production of the whole genome sequence of animals and pathogens. It facilitates the development of toolboxes, high-density and high-throughput genotyping of animals and pathogens. And I'll step back to the previous slide to give you an example. The first bacterial sequence, the chemistry cost about certainly a six-figure sum, and today that can be delivered for about £120. And when the first draft of the human genome sequence, which is similar in size to the bovine genome, was solved in 2004, it took 2 continents, 10 years, and $2.7 billion to produce it. Today, that can be done for $1,000. So it's staggering developments in that field, which are transforming how we do microbiology and genetics. And another technique that flowed from our ability to read the sequence is DNA amplification. A technique that you'll hear a lot about is PCR, the polymerase chain reaction, and variants thereof, which provides, knowing the sequence, you can run small assays which are extremely sensitive and specific for pathogen detection. And just to show you how we have deployed these sorts of tests, I'll start with some applications in virology to start with. Classically, virology was based on isolating viruses in tissue and cell culture or eggs or in some other different media, and then characterizing the isolates by immune responses and other tests. But these approaches have been revolutionized in the last 20 years by the tools of molecular biology, and PCR is now a key AFB technology for detecting viral pathogens, the pathogenic avian flus, blue tongue, etc. And having identified the virus, then it can be characterized by partial sequence. And you would hear on the news that it's an H1N1 virus or an H5N8 virus. So we can provide that level of characterization quickly with molecular tests. And at high throughput, following on from what Joe has just said, AFB is an approved and accredited service provider for the BVD test, which is one of these molecular PCR-based tests. It's a viral load test based on an ear notch sample, and our disease surveillance branch would have done in the order of 1/3 of those tests last year. And the 2 panels on the right illustrate published data where AFB has used these tests to identify the first incursion of H1N1 in European pigs, and providing evidence of horizontal and vertical transmission of blue tongue virus in cattle in 2008. And what's happening is that because of the availability of these rapid sequencing and detection tests, there's rapid global sharing of epidemiological, clinical, and geographic data. And a good example is the global initiative to share avian influenza data. And how that works is if a new virus is identified in China, we don't need to exchange reagents. They upload the sequence, and researchers here and elsewhere can work with that sequence and look at diagnostics and vaccines. And in terms of genomics and virus discovery, again, the classical methods of virology are tissue culture and cell culture, but next generation sequencing has entirely changed how we do virus discovery. And an example of that would be the Schmallenberg virus in 2011 in Germany and the Netherlands. Sequencing of clinical isolates identified a new orthobunio virus, and knew it was that type of virus, but it knew it was a new one. It was named Schmallenberg after the town in which it was isolated. It was isolated from the blood of affected animals and proved to be associated with the syndrome. And very quickly, new tests and new diagnostics were developed on the basis of that sequence. And the bottom panel on the right shows AFBI using tests like that to diagnose or identify the first case of Schmallenberg in Northern Ireland. And how those tests are being developed is they're increasingly being used as part of the horizon scanning approach for the emergence of new virus threats. And I know it sounds a bit like a fishing exercise to just sequence clinical samples, but it is an accepted way of doing virus discovery and a non-selective way of doing virus discovery. Animal vaccines, there's no question, they continue to have a major impact on animal health. The classical approach, using the classical virology approaches of isolation, inactivation, or attenuation, which is passaging, growing the virus in other cell lines or in other species to weaken it. Two world-leading AFBI examples followed the classical approach, and circovirus PCV2 sales are approaching about $3 billion. Salmon pancreas disease virus, again, multi-million vaccine doses annually. Genomics has a role here. Knowing the sequences of these viruses allows us to look at attenuation, because very often the mutations that weaken the virus are not understood, they're not mapped, and genomics provides the ability to do that. And having the genome sequence allows researchers to make what are called subunit vaccines, which are just making the pieces of the vaccine to which the host responds, a technique called reverse genetics, which allows you to make surgically precise genome edits in the virus to make rationally attenuated vaccines based on your knowledge of attenuation. There is the potential there to make multivalent vaccines that provide protection against more than one virus, or chimeric viruses that do the same thing. It provides an opportunity to look at DIVA vaccines that differentiate infected from vaccinated animals, which can you can add in or remove a marker, which allows you to identify the vaccine response and not a wild-type response. So same approaches are appropriate for pathogenic bacteria. The genomes are bigger and much more complex. Currently, we isolate bacteria on specialized media, and you will have seen those spreading bacteria on agar plates. That's a selective approach, and we actually only know how to isolate a very small proportion of the bacteria that are out there. And having isolated them, we run a panel of what are called phenotyping tests, looking at the biology and the biochemistry of the bacteria, like the panel in the bottom left. But there is a transition from what's called phenotyping to genotyping, and AFBI locally uses a molecular test in the last step of TB confirmation. So bacterial whole genome sequencing has the potential to replace that panel of species-specific tests with a more general panel, with a more general platform. And detection of pathogens can then be either specific, when you know the sequence, and selective or non-selective when you don't, and when you're working in the kind of pathogen discovery mode. But the DNA data are fundamentally digital, portable, and more readily compared against international databases, so that you can identify the isolate in these family trees of genetic sequence, and they're much more interchangeable internationally. Genomics and antimicrobial resistance. AMR bacteria could kill in the order of 10 million people a year by 2050 at a potential cost of $790 billion. It's a disturbing fact in the UK O'Neill report, and it's still disturbing that more antibiotics are administered to healthy animals across the world than they are to unhealthy humans. We're currently concerned about livestock-associated MRSA, monophasic Salmonella typhimurium, extended spectrum beta-lactamase E. coli, and other multi-drug resistant Salmonella with high impact in public health. And we're collaborating with with other groups to isolate and sequence these bacteria. Again, pathogen sequencing is likely to have a role here in quickly identifying these bacteria, working out their antibiotic sensitivity, susceptibility, and working out the mutations that are associated with antimicrobial resistance. There are international efforts to index those mutations by whole genome sequencing. And maybe on a more encouraging note, a technique called metagenomics has potential with comparative genomics to help find the next generation of antimicrobials which are badly needed. I'm just going to finish off with a few slides on molecular tests that we have developed from first principles. In AFBI, we have developed a toolbox of genome-enabled tools that allow us to investigate the evolution, epidemiology, and outbreak investigation for TB. And just looking at this particular test, which looks at families of TB strains, we've shown that deep ancestry shows that the UK and Ireland is dominated by their own TB strain family, which we've called European 1, which is not prevalent in the rest of Europe. Zooming in a bit more closely on the actual strains of TB, and these are heat maps for different TB strains, and they show this remarkable local geographic clustering. They're remarkably stable in those positions over time. And it says immediately that the epidemic is strongly locally driven. It is, in fact, a series of micro-epidemics, which is valuable for tracing locally, nationally, and internationally. We make these data available to the veterinary service for outbreak investigation of all confirmed cases of TB in Northern Ireland at the minute. And just zooming in a little bit further, if you let your eyes adjust, we've looked at how the TB strains are between cattle herds and roadkill badgers. The badgers are shown as stars and the cattle herds are circles. So cattle and badger strains are clearly associated at that regional level, which is an important part of the local evidence base. What we were lacking was the direct genetic evidence of transmission chains that link cattle and badgers at the farm scale, and we worked with the University of Glasgow, which is a first in the use of pathogen whole genome sequencing for TB, taking one of those strains, in fact, the little blue one on the inside of the Ards Peninsula, just that strain, the blue one, and sequenced all the isolates we could get. So this is a combination of molecular typing, whole genome sequencing, and mathematical modeling. It provided the first direct evidence of ongoing TB transmission between cattle and badgers at the individual farm level. Again, an important piece of the local evidence base. We also detected signals of cattle-to-cattle spread, amplification, in some of those study herds. So AFBI research is providing insights into disease transmission dynamics which were previously unattainable. And just to finish, I wanted to talk about some work we've been doing on genomics and cattle resistance to bovine TB. A number of parallel studies, including our own, identified heritable genetic variation in risk for TB, which should be exploitable by selective breeding. There are relatively high risk and relatively low risk Holstein sires. And these studies have led to industry-led national genetic and genomic evaluations, estimated breeding values for TB resistance, and these estimates are produced by the Scottish Rural Colleges and AHDB. It's called TB Advantage, a new genetic selection trait launched in early 2016, a world first, and encouraging data from other dairy breeds and beef breeds that they are likely to respond in a similar way to this genetic selection. The Northern Ireland data are being included in these EBV estimates, and AFBI have played a key role in these studies, not least delivering a large case control study where we looked at affected and unaffected cattle herds. For TB, we estimate the heritability of the TB disease trait at about 23%, which is not trivial and should respond to genetic selection. We mapped the genetic variants, and the panel in the top right is a Manhattan plot showing the response, the mutation, the SNP responses, and how they associate with being a case or being a control. We mapped the variants associated with TB resistance, And this study also demonstrated that genomic prediction or genomic selection would be feasible for the TB resistance trait. And that's a new technology whereby you can predict the likely future performance of an animal, from the genotype of a calf. And this is likely to be feasible for TB when the training population is a bit more powerful than it is at the minute. And disease genetics research in the future, only some diseases for a variety of reasons are amenable to this approach, but there are exciting new opportunities to exploit genetic variation in new traits, such as relative infectivity, investigating super spreading, differences in response to vaccination. So just final point, key points. Advances in what the Americans call ag bioscience, or allowing AFBI to undertake things that were inconceivable just a very few years ago. These new tools and the evidence-based approaches are allowing AFBI to help solve some of society's biggest real-world problems: safe and secure food, antimicrobial resistance, animal health. DNA technology is revolutionizing human medicine, driven by the personalized medicine agenda, tailoring medicines to individuals. And similar advances are already rapidly changing the way AFBI does diagnosis and how we understand animal disease. So AFBI is contributing to improved and more sustainable control of infectious diseases in livestock. And in that way, we're supporting local food production and livestock trade. And I'll leave it there. Thanks for your attention.